Hello and good morning to everybody. Welcome to GemPharmatech's monthly webinar series. My name is John. I'm part of the marketing team here at GemPharmatech, and I'll be your host today as you hear from our Senior Study Director of the Immuno-Oncology Pipeline, Dr. Shiying Guo, on the topic Overcoming ADC Resistance, the construction and preclinical applications of Enhertu-resistant models. Before we get started on that, I will briefly explain who GemPharmatech is, in case this is the first time are joining us. As a CRO, our goal is to engineer a healthier world by pushing the boundaries of biomedical innovation. The way we do that, if we go to the next slide, is by providing the tools, the models, data, and insights that move drug discovery forward. We work with academia, we work with biotech, and we work with some of the largest drug and pharmaceutical companies in the world. And we're constantly trying to update these tools, these models, and services to help scientists accomplish their goals. This webinar is focused on ADCs. We work across many modalities as well as many disease indications. So if you're just joining us today, interested in this topic scientifically, but maybe your own work falls into other areas like other oncological areas, autoimmune, neurology, cardiovascular disease, metabolic disease, rare disease, whatever it is, we try to create those tools. And I urge you to follow us on LinkedIn, where we post about those, as well as will you hear about our next webinars and other offerings like that. Some of those tools that I was talking about that are available to scientists, we have the largest collection of genetically engineered mouse models in the world, over 30,000 models. And we have the capacity to generate over 6,000 new models each year. And we work out of 8 locations worldwide, and we complete around 2,000 preclinical studies every year. At the end of this webinar, there will be a Q&A session where you can ask us questions about the content that's being presented. But if you have other questions about the work we do, or if your question doesn't get answered today, feel free to reach out to us at sales@gempharmatech.com. You'll see that link at the end of the presentation. And certainly you can message us on LinkedIn. That Q&A session will happen at the end, as I said, and it will make use of the Zoom Q&A feature. So if you do have a question, I don't know if it's available yet, but by the end of the show, that Q&A section will be available to you. So with that, I'll leave it to Dr. Guo, who will begin the webinar. Thanks again for everyone to join, and I hope you enjoy.
Thanks, John. Hello, everyone. Next, I'll start today's topic. First, we'll explore the construction and validation of an Enhertu-induced resistant CDX model. Next, then we'll move to the investigation of underlying resistance mechanism. And finally, we'll focus on the characterization of a primary Enhertu-resistant PDX model. As we all know, drug resistance is a pretty universal challenge in cancer treatment. It comes from things like tumor heterogeneity, adaptive signaling, and the tumor microenvironment. As this figure shows, even though we are targeting multiple hallmarks of cancer, tumors can still find ways to evade therapy. Understanding how tumors adapt and developing preclinical models with drug resistance is very important for advancing durable therapeutics, including ADCs. So today, we are going to focus on ADC resistance, especially around DS-H201, also named as Enhertu. It is a next-generation antibody-drug conjugate that targets HER2-expressing cancers. By combining a monoclonal antibody with the topoisomerase I inhibitor, it delivers chemotherapy directly into cancer cells, minimizing harm to health tissues. Enhertu has been approved for various types of cancers, including HER2-positive and HER2-low breast cancers, HER2-positive gastric cancer, and HER2-mutant lung cancer. It has shown remarkable clinical outcomes, but like many battles, there are challenges. Clinical data show that about 24% of patients still accept resistance after about 12 months of treatment of Enhertu. To tackle the challenge of ADC's resistance, our platform has developed three Enhertu resistant models, which can be classified into two categories: acquired drug resistance and the primary drug resistance. As for the acquired resistant models, we've established the breast cancer JIMT-1 Enhertu resistance model and its enhanced version, both through in vivo induced method. The GIMT-1 Enhertu resistant is maintained as a form of cell line. It is highly suitable for the drug screening through in vitro cytotoxicity assay. The process will be fast-paced and high throughput. It can also be used for in vivo efficacy evaluation, but a single low dose is recommended because its resistance in mice is not so extremely strong. The next model, the GRMT1 enhanced Enhertu resistant model, is a more powerful version. It was derived from the above model and continuously to be maintained in the form of tumor mass. This model can withstand multiple repeated administrations while still maintaining drug resistance. And it can better simulate clinical drug administration strategies. A third is a primary drug resistant PDX lung cancer model. It was screened through in vivo drug sensitive tests among numerous PDX models. This model can tolerate multiple high-dose administrations, standing out as a prime example of natural resistance to Enhertu. Let's take a closer look at the data of these models one by one. This picture shows the induction process in which GIMT-1 breast cancer cells were inoculated into the mice and developed drug resistance after repeated drug administrations. Currently, this resistant model is preserved in the form of a drug-resistant cell line. From the in vivo validation assay, it is evident that when a single dose of Enhertu at 3 mpk was administered, the tumor growth inhibition in the parental cell reached approximately 63%. However, in the resistant model, this rate decreased to the 32.5%, indicating partially drug resistance. Regarding the cell cytotoxic assay result, it can be observed that the IC50 value of GIMT-1 Enhertu resistant was nearly tenfold higher compared to that of the parental cells. This finding confirms that we have successfully established an Enhertu-resistant cell line. To further enhance the drug resistance level of the GIMT-1 Enhertu-resistant model, we continuously optimized this model and successfully got an enhanced drug resistant model. That is GIMT-1 Enhertu-ER. That means enhanced resistance. The efficacy result indicated here indicated that different from the previous model where 3 mpk dose was used. Here 3 mpk dose was used. We employed 2 higher dose at this time. That is 6 mpk and 10 mpk. Whether in the BBC nude mice or the severe immunodeficient NCG mice, after multiple administrations up to 2 doses or 5 doses, the tumors still demonstrated a very strong drug resistance. The tumor growth inhibition never exceeded 54%. According to our historical data, if this dosage is administered to the parental cells, the tumors will completely regress. I believe many of you will be quite interested in the drug resistance mechanism. Let's discuss this topic right now. According to the reference, the mechanism of Enhertu resistance are very complex. They are involving many reasons, such as low HER2 levels or the truncated forms, defective HER2 internalization, or the increased HER2 recycling, alteration of the lysosomes or the drug efflux pumps related. So we've also conducted some work to explore the mechanism of these 2 models. Firstly, we checked the HER2 expression. Whether detected by flow cytometry analysis or by the IHC analysis, we can see the expression of HER2 in a resistant model was still present. This means the resistance is not due to HER2 expression. Next, to explore whether drug resistance is related to the payload, cytotoxicity AC and IC15 values of different payloads DXD and MMAE were determined for the GIMT-1 Enhertu resistant cells and its parental cells. The IC15 value of DXD in GIMT-1 Enhertu resistant cells was significantly increased, indicating that the cytotoxicity effect of DXD on drug-resistant cells was reduced. When we used another chemical toxic MMAE, both drug-resistant and the parental cells could be killed at low concentration of MMAE treatment. Moreover, IC15 evaluation of another HER2 ADC with non-DXD payload revealed potent cytotoxic activity against Enhertu-resistant cells. These results revealed the resistance of Enhertu in this model is related to the drug resistance of the payload DXD. Our in vivo pharmacodynamic experiments further verified this conclusion. When using another ADC, RC48, which is an ADC drug that links HER2 and MMAE. It can significantly inhibit tumor growth in mice with Enhertu treatment in the resistant models. To get a better understanding of the molecular mechanism related to drug resistance, we conducted second-generation sequencing. From the result, it can be seen that there are a large number of genes being upregulated or downregulated. For example, SLC46A3 is markedly downregulated, suggesting reduced drug internalization or lysosome release, while efflux transporters such as ABCG2 are upregulated, potentially promoting payload efflux. When we conducted a cytotoxicity test of Enhertu on these two cells and added the inhibitors of ABCG2, the KO143 or the elecradar, we could observe that in the group where the inhibitors are added, the ability to kill drug-resistant cells was significantly enhanced, almost returning to the parental to the same level as that of the parental cells. Let's summarize. So far, we have obtained two models of Enhertu resistant in GIMT-1 that acquired resistant tumor models. One is preserved as a form of cell line, and the other is a form of tumor mass. We recommend using the cell line for large-scale in vitro drug screening and use the tumor mass for the in vivo drug resistance verification and new drug evaluation, which can better simulate clinical drugs use strategies. Regarding the mechanism, the resistance mechanism of these two models are not be related to HER2 expression, but are somewhat correlated with the payload and the efflux of the conjugated drugs. These models have conjugates. Okay. Next, I'd like to introduce our patient-derived xenograft model PDX of primary Enhertu resistance. Here, I've listed partial samples from our Enhertu screening on PDX models. As can be seen from this table, for samples with strong HER2 expression, gastric cancers showed a better response than lung cancers. Not shown here, even almost among gastric cancer samples with weak HER2 expression, there is also a good response to Enhertu, which is quite consistent with clinical data. As for the lung cancer samples, we've screened out a cancer case LC 167 number, which has a strong HER2 expression but shows an insufficient response even after multiple high-dose treatment of Enhertu. We call this as a primary resistance. Preclinical evidence suggests that Enhertu resistance may not be limited to a single drug, but can lead to a broader multi-drug resistance. That means MRD phenotype. In this lung cancer PDX sample, we've also observed such MRD phenotype. It is not only resistant to Enhertu, but also show resistance to the antibody trastuzumab and another ADC RC48. Let's take a look at the characteristics of this PDX model. In addition to strong positive expression of HER2, it also has high expression of TROP2 and high expression of HER3. These two targets are also very common tumor antigen targets. So we can potentially try testing other ADC drugs or dual-head ADC drugs in this model. More interestingly, through RNA-seq sequence, we further found that a series of genes related to the metabolic enzymes were significantly and high expressed in this PDX primary resistant model. To the upregulation, such as the genes such as the AKR1 family, that has been reported, which was related to the drug resistance because the high expression of these metabolite enzymes can chemically modify and inactive toxins before they reach their targets. Well, that's all for the Enhertu resistant models and their applications have been introduced today. Actually, we have more R&D work in progress regarding the drug resistant of ADCs. Besides the three models I have introduced today, GIMT-1 Enhertu, GIMT-1 Enhertu resistant enhanced, and the LC00167 Enhertu resistant PDX models. And some another work are still on work in progress, such as Enhertu resistant induced model with another lung cancer cell lines and gastric cancer cell lines. And also we have another target ADCs resistant products such as gastric cancer and breast cancer target TROP2 ADC. Finally, please allow me to use one more slide to go through our ADC platform. It is a one-stop solution for ADC preclinical research, offering a comprehensive suite of services, including in vitro pharmacology study and the in vivo efficacy study. The assays that we can conduct include such as the antibody binding using the flow cytometry or the ELISA assays, and also we can test the antibody internalization using a pH sensitive labeled to the ADC drug. And we can also test the payload release through such experiments such as DNA damage test, a cell cycle, or apoptosis. And in vitro cytotoxicity assay we have introduced in this slide, through mix the antigen positives and the luciferase labeled antigen negative cells, we can also do the evaluation of the bystander effect of the ADC. And as for the anti-tumor efficacy, we have very experienced in this conduction. We can use our CDX bank uh, and to do the orthotopic model using the luciferase labeled tip cells and can using the PDX models. Uh, Besides for that, we have also the capability to do the PK/PD analysis and the non-GLP toxicity test. Make sure that can make sure we can completely comprehensive evaluation of the ADC's performance and safety. Okay, thank you for listening attentively. Now it's time for questions and discussions.
Hi, everybody. Thank you for watching the webinar. As you can see, now is time for our Q&A session. So if you do have a question, please pop it in using the Zoom Q&A feature. Should be towards the bottom of your window on your screen. I'll wait just a moment. If you do have questions that you don't want answered on stream, feel free to send those to sales@gempharmatech.com. And if we don't get to your question today, we will definitely reach out to you with answers to that question. I think we'll kind of take until the top of the half hour here. I will read the first question. Why do you think the second model of the enhanced resistance JIMT-1 and HER2 can better simulate the actual clinical drug use?
Thank you for your question. I think I can answer you this question from two aspects. Yes, we think the second model, that is GMT1 Enhertu enhanced resistant model, is better, which can simulate the actual clinical drug. The first reason is that we can considering the dosage and frequency of the drug administrations, because the clinical Enhertu dosage for major indicators such as breast cancer is about 5 milligrams per kilogram. And for ADC's drug, applying the clinical dosage in milligrams per kilogram of body weight directly into mice is currently the more widely accepted. So, administrated for in the clinical, that is 5 mpk administrated once every 3 week, and repeated dosing. So in the preclinical studies of Enhertu that have been published, the mice were administered a dose of 5 mpk per a tail vein injection every 3 weeks or once a week and continuing several weeks can significantly anti-tumor effect. And you can see in our models, we used the 6 mpk in this enhanced GIMT1 HER2 that was very mimic the clinical dosage, the clinical administration strategy. That is for the breast cancer. As for the clinical dose for gastric cancer, it about the 6 to 7 mpk. So if we want to simulate this scenario, the dose for mice can be increased to the 7 to 8 mpk accordingly. So we have also the 10 mpk dosages in such model. And in such model, we treated them in multiple trees, so they are very mimic. And the second is regarding, I would like to say that is due to its tumor characteristics. Since this tumor is passaged and transplanted in the form of tumor masses, so there are still certain individual differences among individuals. In actual clinical practice, different patients also have a diverse response to drugs. So with this model that has individual difference, we can evaluate the efficacy of drugs more realistically. That is our reason. We call this as a more mimic model to the clinical. Yeah.
Thank you, Dr. Guo, for that very thorough answer. We were trying to limit this webinar to the top of the half hour. So I'd like to thank everybody for participating and see you next time. Thank you very much again to Dr. Guo.
Thank you. Thank you all. Thank you for your time.